So classification of the Rainia here. The class is the Pteridophyta. Order is the Xylophytales. The family is the Rainiaceae. Genus is the Rainia. Okay, clear? Hello. No clear? Okay. So this one is the Rainia is the fossils, which is the representative the Silurian period to Devonian period. These fossils remains are mostly fragmentary and incomplete, so the much of the details in the still unknown. So that's why this Rainia is the renamed after locality of the Rainia in Apprentice district of Scotland. So this is Rainia Pagani, or the better known species are there. What is habitat? There is the evidence that the plants of genus Rainia was growing in the swampy marshes near the Balkans. So here the Rainia is discovered by the old red sandstone beds, means middle Devonian period. Okay. So here the both species of plants were the herbaceous structures are there. Rainia major is the largest species with the aerial stem and measuring around 50 centimeters in height. Okay. Okay. And the other species, that is the Rainia vagani, was the here. In the both species are the known plant were only the sporophyte, which are the rootless, is considered the branched axis. The axis was differentiated into the prostate and are buried under the pitty moss formed in the dead remains of the other plants. Erect and prostate manners are there. The aerial stem was only the continuous to rhizomatous, which is the separated dichotomously branched. There. The stems are cylindrical in structures and unbranched structures are there. So there are the aerial stems was tapering the gradually towards the species the either ended the vegetative apex born in the lateral solitary sporangias are there. The Rainia was the primitive vascular plants. The stele was the simple protostele. With the central core, the xylem is surrounded by the phloem. So that one, there is no pith. The anatomy of rhizome, the aerial stem was essentially same in the both species, means rhinia, that is the so the cross section we can see that one outer circular that is the definition to three regions are there: epidermis, cortex, and stele. The epidermis is the single layer. Outermost layer is the epidermis, which is made up of single layer. A distinct out layer is there, and the epidermis is covered. A cuticle is present. Thick cuticle is there. The typical stomatas are also distributed in the intervals. Okay. So in the rhizomes, the stomata are absent. Next one is the cortex. Cortex below the epidermis, a distinctive cortex is there. The many larger central located in the stele was the differentiated into hypodermis and inner cortex. Okay. So hypodermis consists about 1 to 4 layers. Hypodermis is there and remaining the parenchyma tissue is present. Okay. So, so this is the transverse section of the stem. See here, this one. Okay. Then the inner cortex is mostly probably the photosynthetic tissue is there. Endodermis and pericycle, the separating cortex from the stele, which is the art. The stele is haplostelic protostele is there. So the spiral thickening phloem xylem is present and the smaller diameter peripheral tracheids are also present. Okay. And the phloem is surrounded by the xylem, which had 3 to 4 layer thin wall. These cells are sieve tube cells are there. Sieve cells and sieve plates cells are present. Another one is the reproduction. In the Rhinia, only the sporophytic generation was known. So asexual reproduction, only the asexual in the sporophyte of the Rhinia reproduced asexually by the spores was that one is the homosporous condition. Yesterday we discussed the homosporous means the single types of the spores are there. All spores are similar in structure. That one we call as homospore. So the sporangia which were born on the solitary at the apex of the stems means aerial branches are there. So this is the see here the sporangiospores which is born on the aerial axis. See here, this one is the Rhinia sporangiospores are there. Okay. And the sporangial wall very thick and consists the three distinct layers are there. Outermost layer wall is the stout epidermis and thick cuticle is present. Middle one is considered a very thin wall, and innermost layer is a small rounded cells which are probably function as a tapetum. Tapetum means nutrition food, which is nourished for the development of the spores. So after the spores means complete the development of the spores, there are liberated and to germinate as the gametophytic plants. Okay. So next one is the lycopodium. So this is lycopodium. So they can see here. This is they are the hanging like structures are there. The Lycopodium division is the Lycopsida, class is the Ligulopsida, order is the Lycopodiales, family is the Lycopodiaceae, genus is the Lycopodium. Okay, so the genus Lycopodium. Okay. Uh, Around the 33 species are there in India. So, what is the habitat? The Lycopodium species commonly called as the club mosses. So club mosses means they are found a club-like structures are there in the apical branches. They can grow on the tropical and subtropical areas. Most of the species perfect moist and shady located in the humus is present, where the rich in humus. The habit, which one is the Lycopodium grows and the nature of the sporophyte. The structure of the sporophyte is depending on the species to species diversity is there. So the plants are slender, delicate, weak stemmed structures are there. Okay. Some species, the species without any rhizomes, and some are they have the rhizomes are there. External form. So the plant body is distinct into stem, root, and leaves also there. The stems are either pendant or branched or unbranched structures are the Lycopodium. The first formed root in the short-lived structure. Root system is advantageous system is present. The roots also dichotomously branched. The leaves are Lycopodium, small, simple, and microphyls are there. The leaf gaps are not formed. Leaves are sessile leaves, broad, and the leaf base uh, at the leaf base is broad. They are unbranched, midbeams also present. These leaves are spirally arranged on the stem. Internal structure and growth. If we take the transverse section of the stem here, this is the so in the apical region, some stomata is present. Club-like structures, so that one we call, that's why we call as the club mass. A stomata is present. Okay, and the apical region, that sporophylls are modified as the sporangiospores. Okay, that leaves are to produce the spores. That spills are sporophylous leaves, which are bear the sporangias. That leaves we call as sporophylls. The remaining leaves are normal leaves are there. When we take the transverse section of the stem of the Lycopodium, you can see the distinct structures which is present. This is the transverse section of the Lycopodium. The stem is cross section. We can circular. It is the circular structure is there. Circular structure is there. So which is differentiated into three regions are there: epidermis, cortex, and stele. The epidermis is the one-celled thickness are there. Outer wall is the epidermis, which is the cutinized stomata is the occur in the epidermis. Cortex, which is the several layered cortex, are there very broad and thickness of the cortex is depending on the species. So this cortex is the inner cortex and outer cortex is also there. Below the epidermis, one layer is the hypodermis is also present. And the lowermost layer of the cortex is the endodermis, which is the endodermis is a single layer. Endodermis is there. So in these endodermis have the Casparian strips also present. This is the distinctive character of the stems of the young stems. So another one is the below the endodermis is the stele is present. So the stele is consists around 3 to 6 layers of the rectangular cells periphery forms. That one is the pericycle is there. So inner the pericycle, the stele is considered solid core. Xylem is centrally, which is surrounded by the a ring-like phloem is there. Xylem is centrally present. There is no pith, so that's why we call it a protostele. The Lycopodium is the protostele condition is there. So depending on the species, the stele is different types are there. Haplostele, actinostele, pectostele, and these types are there. So what is the haplosteles means? So only the xylem is surrounded by the phloem centrally. So actinostele. So actinosteles means the stele is to actin-like structures are there. Okay, actin-like structures. So that one is called as actinostele. See here, this diagram. There is the different types of the steels are present in the lycopodiums. This one. This is the actinostele, and centrally the xylem is surrounded by the phloem. Actinostele, plectostele, and mixed protostele. Like the structures are there. Okay. So the next one is the leaf structure, the reproduction. Next goes to the reproduction here. The sporophyte of the lycopodium is reproduced by the vegetative asexual methods are there. The vegetative reproduction means the growth, the and death of the older parts of the rhizomes and some tuber-like structures also there, and gemmas and buds also develop from the aerial part of the lycopodium. Okay. Another one is the asexual reproduction. The sporophyte is the homosporous condition is there. Homosporous means all the spores are single type of the spores are there structurally, morphologically, and physically. So that's the homosporous condition. Lycopodium is the homosporous. And it is reproduced asexually by the function of spores. The spores are formed in the sporangia of the axial. The sporophylls appear in the different from the vegetative leaves. So these sporophyll leaves are in the apical regions are there. Uh, they are called as the stobalus like structures are there. In the previous slide, we can shown that one. This one is the stobalus. Stobalus are nothing but chlorophyll leaves. Chlorophyll leaves are there. Club-like structures are there. Stobalus, this one. Okay. These sporophylls are very different from the vegetative leaves are there. The sporophylls are just like a sterile leaves except their small leaves. There are the sporangia are compactly large and kidney-shaped structures are there. At the maturity, they are yellow in color. Each sporangia so they can show the multicellular stalk and capsule is there. So this one is the vertical section of the strobalus. See here, sporophyll. This one is a sporophyll. On the sporophyll, adaxial, they have a sporangia. Adaxial area means upper part of the. They have a kidney-shaped sporangias are there. This is the sporangium. Inside the sporangium, number of spores are there. At maturity, they are yellow in color. Each sporangium shows the multicellular stalk and capsule is there. The wall of the capsule is three-layered in thickness, with the innermost layer is the function as a tapetum. So, so the inner tapetum spores to development of the sporangium is eusporangiate structure is there. A single row of the superficial cells of the adaxial surface of the sporophylls near the base of the function as a sporangium initial. So that is the spores are applied in conditions are there. The very small in structure. There are the 0.03 millimicron to 0.05 millimicron in diameters are there. They are in the tetrahedral structure is present. These spores are coated in two layered in thickness are there. The outer coat is the thick is showing the reticulate ridges like structures, slopes like protrusions and lines ridges are there. The inner one is the smooth layer is there. Here, the gametophyte and sexual reproduction here. That is the first type, second type, different types also involved in the reproduction system of the gametophyte here. So the Anteridia, which is the developed are either completely embedded in the slightly projections. It is very simple structure. Mature Antheridium is consist of the body with a single layer of the walls and closely large number of spermatozoids also there. Inside the Antheridium, the spermatozoids are there. It is divided transverse into the two cells. The outer cell was the jacket initial, which is undergoes to the several anticlinal divisions to form as a antheridial. So after the antheridium initials, the androgonial cells also there. These androgonial cells to undergoes the number of division to form the androcysts or spermatozoids. So each spermatozoid they have the flagella. They can swim and to reach the egg to form the zygote. So Archegonium, the Archegonium, this one is also sunken with the their neck projections from the gametophyte. They have the egg and the ventral canal cells and neck canal cells also there. So after the fertilization, they can develop as the. So here the development of the.
Hello.
So after the embryo, wait. After the embryo, see here the after embryo will be developed in the different types are there. That is the see here that is also to the Mm. After fertilization, the egg is after getting the itself surrounded by the a wall divide transversely, so which is to long axis of the Archegonium and enlarges to form the suspensor. It may be remains undivided or divide sometimes to form the 2 to 3 cells. So that is the hypobasal cells, which is called as embryonic cells, undergoes the two vertical divisions and transverse division to form the 8 cells. At this stage, the embryo consists of the 1 plus 8 cells. So upper quadrate go towards the suspensor largest and subsuperior foot or the itself into the gametophyte tissue. Hmm. So, and the second type is what happened here. The development of embryo into 1 plus 8 cells are there in the similar in structure. Further development shows variations are there. In the upper 4 cells are foot and lower four to form the undifferentiated mass of the globose structures are there. That one may be called as protocom. Okay. It became the green and produced the rhizoids from the lower surface. They have the stomata and function. Some leaves also there means protophylls also developed from these protocol. So here the tube means in 1890 they have the established in the protocom theory is developed. This protocom is the structure of the great antiquity and regard the precursors of the stem. Means precursors means initial product. So that one, this view of the opposite by the staining, that is the protocom was unlike and sporophyte in the absence of the root and vascular tissues. Okay, so this is the life cycle of the Lycopodium. Next one is the equisitum. So equisitum, this is the this is the equisitum. So this is also so this equisitum is division is the Phenophyta, class is the Calamapsida, order is the Equisitales, family Equisitaceae, genus Equisitum. The plant body Equisitum, which is a sporophytic plant body, just like the Lycopodium. Okay. The type of the Equisitum is included around the 25 species are there. It is very rough texture is there. We can feel that plant which is very rough in texture is there. Became which is a species of poorly known as a horse tail. Just like plant is looks like a horse like structures, tail like structures are there. So the genus is cosmopolitan distribution is it is found overall the world except Australia and New Zealand. Then structure of the sporophyte. So habitat, the ecosystem, there are thrives in varieties of habitat as there. There is the urban grows and exposed to located locations that is either in the grasslands and wetlands, some sandy sites also, sandy roadsides also they can. So they can grow on the pond and marshy locations and river banks also. So what is the habit? Means there are the annual perennial herbs that are differentiated into stems, rhizomes, and leaves also present. There are the slender and generally creeping like structures are there. Okay, creeping like structures are there. There are the height around the 3.5 to 4 centimeters in height. So the sporophyte ecosystem is distinct into stem, leaf, and roots also there. It is conspicuous by the articulated stem and hold some scale-like leaves also. The scale-like structures that one we call as the leaves. They have the nodes and internodes. So the stem is have nodes and internodes. So at the nodal region, these scale leaves are appear. The rhizomes or stems grows horizontally. Sometimes is penetrate into the depth in the, 1 meter under the soil surface. Each node, a whole scale-like leaves are present. They are the laterally fused towards the base of the base to from the brown sheath around the node. So alternating the leaf is branch primordium is present at the each node of the rhizome. So branch primordia develop into the either subterranean or aerial branches or many dormant over the loops period. One is there. So we can see the structure of the stem. The stem is see here, transverse section of the stem. This one is the stem transverse section. Okay. The stem of the ecosystem is naturally cladoed. Cladoed means the plant total involved in the photosynthesis. Means the pigments also there, chlorophyll pigments also there. So that's why the stem is involved in the photosynthesis. They are totally green color. So this one. So a separate unbranched fertile branches also there. Sterile branches and fertile branches are there. Okay. So these fertile branches bear the a solitary terminal stabile is there. These fertile branches are non-green in color, means there is no pigments. So that's why not doing the photosynthesis. Fertile branches means they have the bear the stobalus. The branching is die up after the dispersal of the spores. In some species, the fertile branches through the stabili after the liberation of the spores and function like the sterile branch. So the leaves are small in structure. Leaves are small in structure, microphyllous and scale-like structures are there. Number of leaves are present at each node. So there are the cover the between 3 to 40 scales are spirally arranged on the nodal region of the stem. So roots are advantageous. Root system is present. Usually they are found at the node of the rhizome. They develop from the base of the branches primordia at the each node. We can see the transverse section of the stem. See here, the aerial sterile branch is circular but wavy outline is present in this ecosystem stem. Wavy like structures are there. That outline is the wavy like structures are there. So the stem is differentiated into 3 regions are there: epidermis, cortex, and stele. The epidermis is which is consists of single layer cells on the outer wall of the epidermis are very thickened with the silica deposits are present. So that's why we can feel that one which is very rough in texture is present. Okay. So this is the fine granules and rosette or spines. They are also there. The epidermis is also provided with a thick cuticle and stomata. Each stomata consists of two pairs of the cells are there, that is the guard cells are there. The inner pair of the guard cells are compactly over arched by the pair of the subsidiary cells. Okay. So this stomata means to exchange the gases. So below the epidermis, cortex layers are there. Cortex is broadly highly differentiated in the function of photosynthesis and mechanical support also. The cortex is differentiated into hypodermis, mid cortex, and endodermis. So hypodermis is the below the epidermis, which is 4 to 5 layers of the hypodermis is there, which is made up of the sclerenchyma. So that's why they can give the mechanical support to the that axis. Next one is the middle cortex. That is the middle cortex is made up of the parenchyma tissues and chlorenchyma tissues also there. And innermost layer of the cortex, that one we call as endodermis. So these endodermis, they have the transparent strips also present. So below the endodermis, you can start the vascular tissue, vascular canal. So these the middle cortex, some pores also present. That is the vallecular canals also there. That is the middle cortex, some portion inner hypodermis all consists. So air chambers, that is the prominent air chambers are there. That one we call as a vallecular canals. So these vallecular canals are present as a ring in the cortex. Ring-like structures are there. And the endodermis is varies with the species to species. So steel is the siphonosteel. We can consider the steel is siphonosteel is present. It is consist of a ring of vascular bundles are there. The number of bundles are collateral and endark condition, which is resemble to the vascular bundles of the ring of the monocotyledon stem. Just like a monocotyledon stem, which is the similar in structure of the ecosystem. Xylem is fully developed. Each vascular bundle consists of two metaxylem points and widely separated to located toward the periphery face. So here the xylem is also consists the carinal cavities also there. So these carinal cavities are the lysogenous cavities. That one is the protoxylem is the lysogenous cavity. That one we call as the carinal cavities. So this one contain the water. Vascular canals, that is the vallecular canals contain the air, but carinal canals are consist of the water. Okay. So next one is the root anatomy. This also root anatomy is the cross section. We take that one. It is different into three regions also there: epidermis, cortex, and stele. Epidermis is a single layer like that stem. These barrel-shaped structures are there. So these epidermis bear the root hairs. So that's why that one we call it the piliferous layer of epidermis is there. Another epidermis below the epidermis cortex is there. That is the 3 to 4 layer thickness is there. Sometimes it is the outermost layer of the maybe thick wall. It is appeared as the hypodermis means ectodermis or hypodermis. So initially endodermis is a character distinct double layer structure is there. This is the fifth character in the equisetum. Double layer endodermis is present, and innerly pericycle is also there. The stele is centrally stele is considered. So the small cortex stele is there. Next one is the reproduction here. So the reproduction here, the vegetative and Anteridia and Archegonium development. So vegetative asexual reproductions are there. The vegetative reproductions means they are propagated to the tuber which are only in the specialized short branches are there. So some branches are so these tubers are developed as the new uh, ecosystem plants are there. Another one is the asexual reproduction. In the asexual reproduction, they are bear the sporophyte means sporophylls also there. That is the ecosystem is also homosporous condition. They are look like a Lycopodium means the stabales are solitary or terminally they are there. That is the sporophyll leaves. Each stabales is a spindle-shaped structure which is consists of central axis and bearing the several uh, sporangiospores are there. So these sporangiospores are have the sporangium. Okay. So these sporangiospores are projecting like their central axis are there. The sporangios are borne on the rays around the stalk. They are compactly arranged. The pelted disc provide the protecting cover for the sporangia located underneath. Okay, so that is the all spores are here. Homosporous conditions are there. The spores are liberated and they are spherically and uninucleate condition is there. The spores will within the young stages, but they gets complicated after means the spores will show four layers are there. Innermost layer and endospores is thin and consists of the cellulose. Outermost is the endospores, exospores, and middle layers are there. So after these spores are germinated and to develop as the gametophytes. In that gametophyte, they bear the reproductive organs are there. That is the the Anteridia and Archegonium that one developed. See here the development of the Anteridia and Archegonia. See here, this is the structure is the Archegonia. Development means Archegonia initial is undergoes to the pericanal division. That is the central canal cells and cover cells are there. Again, these cover cells undergoes to the vertical division to form the two neck canal cells. Then ventral canal cells are to have the swollen based and ventral canal cells are there. So in that column based egg and ventral canal cells are there, neck canal cells long, slender like flask like structures are there. Okay. So so when their anthrozoids are liberated from the mm, this one, this is the androcyst means antheridia is there. The antheridia initial is there. That antheridia initial again to formed as a jacketed anthrozoids are there. These anthrozoids liberated to fuse to form the zygote and the zygote to develop as sporophytic plant body. Okay.